Plasmids are considered transferable genetic elements or replicons. They are actually naked DNA. Plasmids are important tools in biotechnology and genetic labs because they are commonly used to multiply or express multiple genes. Plasmid is a double-stranded circular extrachromosomal DNA of bacterium. It is used in recombinant DNA experiments to clone genes from one organisms and to make larger quantities of their DNA. Plasmids can be transferred between the same species or between different species. Plasmid characteristics. Size of plasmids range from 1 to 1000 kilobase pairs. Even the largest plasmids are considerably smaller than the chromosomal DNA of the bacterium. Based on functions, they are classified in 5 types. Fertility plasmid for conjugation. Resistant plasmid, which contains genes that probe resistance to antibiotics. It also helps bacteria in producing pilus. Cold plasmid, which contains genes that code for bacteriocin. Degradative plasmid, which help the digestion of unusual substances like toluene. Virulence plasmid, which is responsible for the pathogenicity. Also, there are different structures. Naked open circular DNA, which has one strand cut. Relaxed circular DNA is fully intact with both strands uncut, but has been enzymatically relaxed. Linear DNA has free ends, either because both strands have been cut or because the DNA was linear in vivo. Supercoiled DNA is fully intact with both strands uncut, and with twists built in, resulting in a compact form. Supercoiled denatured DNA is like supercoiled DNA but has unpaired regions that make it strikingly less compact. This can result from excessive alkalinity during plasmid preparation. When a bacteria is transformed with a plasmid, it acquires new characteristics. For example, the transformed bacteria becomes resistant to particular antibiotics, resistant to heavy metal, production of a large amounts of proteins like insulin, ability to transfer the plasmid to other bacteria, production of bacteriocin, codification of colonization factors to invade the tissues of a host species, change metabolic pathways to use different carbon sources, generation of tumors in plants, establishment of symbiosis with plants, biodegradation of toluene and xylene. Plasmid purification. DNA yield depends on the quality of the cell lysate used. Preparation of a chloride cell lysate is therefore a critical step in the purification procedure, which has been carefully designed to provide optimal lysis conditions. The bacterial cells are lysed on NaOH SDS in the presence of RNase. SDS leads to lysis and the release of the cell contents, while NaOH denatures the chromosomal and plasmid DNA as well as the proteins. This denatured form of the plasmid runs faster on agarose gels and is resistant to restriction enzyme digestion. The lysate is neutralized by the addition of acid potassium acetate. The high salt concentration causes KDS to precipitate, and denatured proteins, chromosomal DNA, and cellular debris become trapped in salt detergent complexes. Plasmid DNA, being smaller and covalently closed, renatures correctly and remains in solution. Separation of plasmid from chromosomal DNA is based on the coprecipitation of the cell wall-bound chromosomal DNA with the insoluble clusters containing salt, detergent, and protein. Plasmid DNA remains in the clear supernatant. Vigorous treatment during the lysis procedure will share the bacterial chromosome, leaving the free chromosomal DNA fragments in the supernatant. RNase-A, which is added at the beginning of the procedure, digests the liberated RNA efficiently during the alkaline lysis. The precipitated debris is removed by centrifugation. It is important that the lysate is clear at this stage to ensure a good flow rates and, ultimately, to obtain protein-free plasmid DNA preparations. Differences with other nucleic acids. For genomic DNA, the lysis is considerably easy, because all that is needed is a good and strong lysis to release the DNA. Then, the purification can be done with phenylchloroform or with other simple methods. For the plasmid DNA, the process is different. It is used an alkaline lysis. The lysis buffer contains sodium hydroxide and SDS, the purpose of which is to completely denature the plasmid of the genomic DNA. Then, the sample is neutralized with potassium acetate to renature the plasmid, and this is the key to the separation of the plasmid and genomic DNA.
First of all, the bacteria with the plasmid are grown in algae medium overnight. After this, they are centrifuged and lysed with a lysis buffer that contains detergent like SDS and a strong base like sodium hydroxide. The detergent pops holes in the cell membranes. Sodium hydroxide loosens the cell walls and releases the plasmid DNA, and also denatures DNA, linearizing it, separating the strands. This is incubated, and a step where potassium acetate and acetic acid are added is performed. The first is to renature circular DNA and to precipitate single-stranded DNA, and also to allow the easy removal of SDS. After the centrifugation step, the supernatant is transferred into a new microtube to precipitate the plasmid with alcohol and salt. A spin is performed and another wash with ethanol 70 is made and let dry. The final product is resuspended. For better results, RNAs may be added to avoid the contamination with this molecule. You must take into account the type of bacteria you are working with, as gram-positive bacteria have a thick peptidoglycan layer in their walls. This characteristic often makes difficult the plasmid extraction, so lysozyme is added. This protein will break the carbohydrate chains in the wall, destroying the structural integrity of the cell wall and cause the bacteria to burst under its own internal pressure. Lysostafin may also be used. This protein is added with the SDS sodium hydroxide solution to lyse the cells. Plasmids can exist as supercoiled, open circular, and linear forms. Normally, they are supercoiled to fit into the cell, and it is expected to obtain this form with extraction protocols. As it is a compact knot, it generates less friction with the agarose and therefore is able to travel more within the gel, generating the last band. With the cut of only one strand of the plasmid, an open circular form is obtained. It will generate more friction and travel less in the gel, generating the first band. With a double excision of the strands, a linearized form is obtained. It will generate less friction than the open form, and it's identified by the middle band. From the obtained results, no isoforms were able to be identified, as RNA presence is observed in both lanes. No proteins are observed in the wells, also plasmid degradation is present. Three ranges can be identified where the isoforms should be present. The relaxed form is expected at almost 2000 BP, the linear isoform between 1500 and 2000 base pairs, and the third one between the 550 and 650 base pairs.
The steps for an effective extraction of plasmids are the following. Centrifuge for 20 seconds to maximum speed to concentrate the bacteria into a pellet, then remove the supernatant. An optional step for a cleaner pellet is to wash it with STE buffer and repeat the centrifuge process. After that, resuspend the pellet into saccharose solution using G2E and adding lysozyme to break the membrane of the cells. Then it is added another lysis solution of SDS and sodium hydroxide. It is needed to wait 5 minutes to let the lysis of bacteria to be completed. After this, to neutralize the solution, it is used high salt solution. Now, since the plasmid will be in aqueous phase again, it will be used 2 volumes of absolute ethanol to precipitate the plasmid. To help this process, centrifuge and remove the supernatant. For a final step, use RNAs to remove the RNA and leave just the plasmid DNA. When a plasmid DNA is extracted without impurities and run in an agarose gel, it is possible to observe three main bands. From bottom to top, the first one will be the superhelical form of the plasmid, the second the linearized form, and the third one of the relaxed circular form. In the left image, it is possible to clearly observe these three bands. On the other image, which is the one obtained in the laboratory, it is possible to see the same three bands, although not as clear as in the other image. The results after the electrophoresis indicate us that the plasmid have a good purity because even though it is observable a degraded band of RNA in the bottom, it was not used in the protocol RNAs, so this is normal. On the other hand, as said previously, some of the bands can be clearly seen. This might be for a low concentration of plasmid, except for the superhelical form in which has a greater concentration. However, this last band has a little bit of degradation. To make an alkaline lysis, there are four major steps. The first one is to obtain and clear a pellet of the bacteria using centrifugation and STE buffer. The second one is the lysis of the membrane to obtain the plasmid using lysosome, SDS, and sodium hydroxide. The next step is to neutralize with a high salt solution, and finally to obtain the pellet. A good electrophoresis of a plasmid DNA must result in three observable bands for its different isoforms. In the electrophoresis done in the laboratory, it was possible to observe these three bands, although two of them with a low concentration, which were the linearized and relaxed circular form.